Okay, can somebody confirm you can hear me?
I can hear you.
Great. I had trouble logging in because the Zoom link isn't there in in Canvas, and that's how I logged in to get to the lecture. Somebody have a comment? And so I had to go into Zoom, and to verify, they want me to log in with Clark, and then Clark wants me to verify my existence by sending it to my phone, and then I check the email and something, and then I can get in. But like I said, this is just really a pain. All the security steps you have to go through to get things to work now. Um, any questions before we begin? My computer is going to be a little slow because it's still making the lecture file. How do I word that? It's still making the lecture file that I will put on the post on the web page, meaning the Zoom file, when it's made, isn't useful to anything but Zoom. And you have to make it into a page that can go into the internet. And it's only about 50% done. So this will take up most of my computer's resources. And my computer will be slow until that finishes. I suspect it'll be slow for the next 10 minutes or so. All right, any questions before we begin? If not, let's begin. I took a cough drop after lecturing, so um, let me crunch it, and then it'll be out of the way. Give me a minute here. I'm trying to get things together. All right, there we go. So, are there any questions before we begin? If not, let's begin with the syllabus again. I don't think I'm showing my page. All right, you should be able to see my page now. So this lab will be budgeted from 7 to 9:20, but I will only talk for maybe the first half hour or so. And I'll give you some pointers on doing the lab and the main part of the lab. But I will then turn it over to you guys to work on the lab. And you can ask me questions, but you don't have to. And after I'm done lecturing, you can close down the browser to the lab as well. And I will stay here until the last student leaves or until 9:20. I usually in this class, students leave when they get the lab done, or some students just leave and get the lab done at another time. And when all the students are gone, I usually close down, which usually happens around 8. Oh, good. My page for the lecture just finished, so now my computer will start working faster. So it's budgeted to 9:20, but in reality, if you have a question, you should log in early, like the first half hour to the next half hour at the very least, because sometimes students log off uh, around 8 o'clock. And if all the students are gone, I'm not going to stick around. Any question about any of that? All right, let's go over a little bit of the syllabus that we need to cover for the lab. I've already talked about the grading, so I'm not going to talk about the grading. The grading and the lecture and the lab, they are two separate courses, but the total grade will be added together. And so the total will be out of 720 points. And so most students don't treat it as a separate class, but you can if you want. I guess I should shut my door in case my roommate comes out. I opened it for 10 minutes to cool down the room in here because the air conditioning is out in the main part of the place, not in here. Ah, let's talk a little bit about microscopes. You're not going to be working with an actual microscope in the lab, but we will have a virtual microscope that acts like a microscope online. So you will be using an online version of a microscope. You are expected to know how to use a microscope from the microscope lab. And I think at this point, that's all I want to say. But we don't actually have a microscope you will be working with, but you will be working with a virtual microscope. Safety information for students and physicians. Because this is an online class and the lab will be done online, you will not be exposed to any live microorganisms in the class. We will pretend that you have microbes that you're growing, but everything will be done online, so everything will be virtual. Is there any question about any of that? This information is for safety information for students and physicians, and it's something you should know because you've taken microbiology, but it isn't really of importance because you're taking an online class and the lab will be done online. You should know that everything in the lab will be either a biosafety level 2 microbe or a biosafety level 1 microbe. A biosafety level 2 microbe person must wear protective equipment like a lab coat, gloves, goggles, and must wash up both before and afterwards. Students who are immunocompromised, living with or caring for an immunocompromised individual, a student who is pregnant or may get pregnant, would not want to work with a biosafety level 2 microbe because they can cause illness. Okay. And alternate accommodations would be made if we were actually coming into a real microbiology lab. The other organisms we will be working with are biosafety level 1, and these don't have much personal risk. You can get sick if you were to consume them, but generally these organisms do not cause disease, at least when they're growing in the normal place for the organisms. Like E. coli grows normally in your gut, and as long as it's in your gut, it does not normally cause illness. But it can cause illness when it's growing in other places, like in the lungs, it would cause pneumonia. In the ear, it would cause an ear infection. Any question about any of that? Anyways, biosafety level 1 organisms are the lowest risk, and some would be immunocompromised. We would give the student these organisms to work with. We wouldn't give them a biosafety level 2 organism. You don't really need any protective gear for a biosafety level 1 microbe. You should wash up beforehand and afterwards, but that's all you need to really do for a biosafety level 1. Biosafety level 2, you should take precautions. We call them a medium risk organism, but we give those to students because they are not, I don't know, a dire human pathogen, something like Ebola. Okay. You wouldn't die normally when you were to get Shigella, dysentery, or Staphylococcus aureus. You may need antibiotics and you may get an upset stomach or your skin may get irritated, but generally you would not die from these biosafety level 2 organisms. So I'm just going over a little bit of the lab safety rules. Lab safety rules will be described in the first week, and that'll be actually our first lab. We're not going to physically meet in a microbiology lab, but you do need to know about the lab safety protocols for a microbiology lab. And many nursing programs report that safety is one of their main skills that they want their students to learn. Safety is of importance in the lab, and your attitudes and actions will determine the level of safety. You should have a positive and respectful attitude towards microorganisms. Never take shortcuts. Use the procedures that you've been taught. And if you do, you will minimize your risk as well as the risk of everyone in the lab, as well as the accuracy. All right, you're not normally allowed to take any personal items to your lab bench. All personal items must be remained in your bag, like your book bag or what, which is in a cubbyhole, which is a little shelves, like little closets where you put that stuff in. And when you walk in the door of the microbiology lab, the door and what I call the cubby holes, the little shelves that you put your things in, are considered outside the lab. And then you walk a few feet further, you're now in the lab proper. Okay. And everything in the lab proper should be something that can be exposed to a microbe and should not allow the microbe to leave the lab. So, like, you can bring a pencil into the microbiology lab and use it in the lab, but it must remain in the lab. And it can never leave the lab until it has been sterilized, either by putting it in disinfectant for 15 minutes or put it in an autoclave to sterilize it. We'll talk a little bit more about an autoclave in a little bit. So all personal items must be in the cubbyhole, and that doesn't include personal items that you're wearing. Like, you can bring in your shoes, you can bring in your shirt and your pants, you can bring in jewelry that you're wearing. We do ask that you don't touch these items so that you would contaminate them. But you can bring in anything that you're wearing. Anything that you're not wearing should be left in the cubbyhole or the cabinet by the door. And that is considered outside the lab. Any questions about any of that? So what do you do for notes? We provide paper and pens and pencils in the lab, and these always stay in the lab. And if you want to take your home notes home to study, we have a what do you call it? An Apple laptop to take a photograph of your notes, and then you can email the notes to yourself, and then you can get your notes and study at home. But everything that comes into the lab proper, meaning everything beyond the door and the cubbyhole, must not ever leave the lab. And if it comes into the lab, it stays in the lab until it has been disinfected. So if you want to take the pencil home at the end of the term, it has to be disinfected before you can take it home. Any questions about any of that? So phones and other electronic devices are not allowed in the lab. You cannot use a phone. And if your phone were to ring, you would have to first take off your glove, take off your lab coat, go wash your hands, go answer the phone, take the phone out of the lab door, we'll call it that way, answer the phone, come back into the lab, put away your phone, and that's still considered outside the lab because you're putting it in the cabinet or cubby hole. Go wash your hands, go put on your lab coat, and then go put on your gloves. Any question about any of that? If a phone or something were to be prohibited in the lab, it would be confiscated and you would not get it back until it had been either disinfected in disinfectant like bleach for 15 minutes or put in the autoclave to sterilize it. That would destroy your phone and any electronic device. Well, maybe a waterproof watch could survive the disinfectant, but everything else would be destroyed. Now, if you're wearing the watch, you may take it into the lab. All we ask is you don't touch the watch while you're in the lab. You can look at it, get the time, but don't touch it. Any questions about any of that? The same with your jewelry. You shouldn't touch the jewelry that's hanging from your ears or around your necklace. And that's for safety. Anything you bring into the lab should not be contaminated with microbes, so then you could take those microbes home with you. So you are allowed some personal items into the lab as long as you're wearing them. The main ones are jewelry, watches, and obviously your eyeglasses. But people who have hearing aids, they're allowed to take those in too. All cultures you're working with should be considered potentially pathogenic. Keep your hands, pencils, and other materials out of your mouth, your ears, your nose, etc. No eating or drinking, smoking, or gum chewing in the lab is allowed at any time. You are required to wear gloves, which are provided for by the lab. You should also monitor yourself for symptoms of infection like diarrhea, fever, abdominal cramps. And if that happens, obviously call your physician and let them know you're in a microbiology lab and whatnot. And you can give your physician the 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 document on microbes and possible contamination. It's called Information for Students and Physicians, provided on the class website. You must wear proper attire in the microbiology lab. You must wear closed-toed shoes at all times, sandals, thongs on your feet, and women's fancy shoes that are open-toed are not allowed in the lab. You may wear them to the lab and then switch shoes to a closed-toed shoes while you're in the lab. And then you go and wash your hands, put on the lab coat, put on your gloves. The lab coats are generally provided for you, but students can buy a lab coat from the bookstore and then bring it into the lab. And that's the last time you will be allowed to bring it in. And then that stays in the lab until the end of the term. If you want the lab coat, you let your instructor know and it'll be set aside. It will be autoclaved, washed, and dried, and then hung up for you to come and get after the term ends. Any questions about any of that? You must wear goggles anytime you're working with a broth, a liquid culture, or when a splash hazard exists. That's generally when we're working with stains and we're staining a slide. So a liquid culture or a stain, you must wear goggles. Goggles are supplied by the lab and they're sterilized in the lab. So after you use them, they'll be sterilized by UV light. Failure to wear goggles may lead to a loss of points, which obviously we're an online class, so you don't need to worry about that. Cleanliness is a virtue in microbiology, and even though you're not going to go into a real lab, you must know how to clean a real lab, microbiology lab. So when you come in, like I said, you first would wash your hands, and you do it for 20 seconds, washing everywhere. And then you dry your hands, you put on your lab coat, you put on your gloves, and then you go to your lab bench. And before you begin working, you disinfect the lab bench to sterilize the area before you work. That will get rid of any microbes on your bench that may have been spilled by a previous student. And let's say they forget to clean up afterwards. And then when you're all done working for the lab, you once again disinfect your lab bench as right before you leave, and then go and take off your gloves, take off your lab coat, go wash your hands, grab your books and things from the cabinet or the cubbyhole, and then you can leave. Any question about that procedure? So you must wash before you begin work and then after you're done working. In a normal lab, you should note where the fire extinguisher or fire blanket, eye wash station, a first aid material are located. In this microbiology lab, you just must know, this online lab, you just must know that in a real microbiology lab, these safety equipments would be in the lab. And you should know what they are, and you don't need to know where they are, but you should know that they are in the lab for your use in case you need them. Once you have an accident must be reported to your instructor. Minor accidents should be cleaned as follows. Always use a brush and a broom and a dustpan to pick up broken glass. Never use your fingers, even if you have gloves, because we do not want you cutting yourself, and then broken glass can easily cut a hole in your gloves and then contaminate you. Spilled strong acids and bases should be neutralized and wiped up quickly. Spilled bacterial cultures, meaning something that has live microorganism in it, should be covered with paper towels first, and then you drench the paper towels with disinfectant. You let it sit for 15 minutes before wiping it up, and that's to kill the microbes. And then you clean it up, removing the broken glass present first with a dustpan and a paper towel. The broken glass goes in the broken glass container. I forgot what you call that, but we'll just call it a broken glass box, a receptacle. And then everything that had contact with the culture would be wiped up with dry paper towels and put in the biohazard bag. The biohazard bag I also call the autoclave bag, and that would be autoclave before it is disposed of in the regular trash. And it is autoclave to kill all the microbes. Any question about any of that? Never put glass in the biohazard bag. Never put glass in the trash can either. Broken glass should go in the broken glass container. And if you have broken glass, like test tubes, that you are done using, you should put it in the test tube rack for used materials. And what that would be done is it'd be autoclaved, and then it would be washed, and then it would be reused in the microbiology lab or some other lab. So you must be sure your hair and your paper are kept away from Bunsen burners and back to incinerators. If you have long hair, it must be tied back. And we do have hair ties in the lab if you do not have a hair tie. Gas back to incinerators, as well as the back to incinerators, should be turned off before you leave the lab. And generally, if you're not going to be using them, they should be turned off and then turned on the back to incinerators about 5 minutes before you use them. The Bunsen burners can be turned on right before you use them. Mouth pipetting is not allowed. Never take cultures out of the labs. Samples of lab materials cannot be brought into the lab unless it is authorized by the instructor. So don't bring any microbes into the lab unless you clear it with me. And that would be if we went into a real lab. Obviously, I don't think you'll be doing that in an online lab. If you have a cut or a sore on your hand, you should wash your hands the normal way, then go to the first aid kit and apply a Band-Aid over the wound, and then put the glove on top of the Band-Aid. And then when you leave, you take the glove off, go wash your hand, and if you want, you can put on a fresh Band-Aid. All microbiology cultures must be completely labeled in the lab with your name, the date, the nature of the specimen, and the medium used. You may also have, it's optional to put the temperature you're growing it at. So all cultures in the lab must have your name, the date, the nature of the specimen, meaning what it is, and then the medium that you're using. And then optional, you may put the temperature that you grow it at. The nature of the specimen would be, if you know what it is, you put E. coli. If you don't know what it is, you can put something like air exposure. Old specimens must be discarded properly in the following ways. Agar plates should be discarded in the appropriate labeled trash cans, and we call those the biohazard container. Glass slides and cotton swabs should be put in the glass container containing disinfectant that's found at the rear of the lab. And then for swabs, we also have one container on each table for putting swabs. And there's disinfectant in the discard swab container. Glass test tube should be labeled, have the label removed, like if you put tape on it to say what it is, remove that, and then place it upright in the test tube rack in the discard bin in the rear of the lab for glass test tubes. Remember, no glass ever goes in the autoclave bag or in the garbage can. If you have broken glass, you can put it in the broken glass container. And we don't really use glass bottles or birettes. But if we did, you'd put that near the glass test tubes. We do use glass test tubes. We do not use glass pipettes, but if you had glass pipettes, you'd put it in with a glass pipette discard pile, not discard, but reuse pile. And we only have plastic pipettes, in which case you put plastic pipettes. If you don't have contamination, like you measured water in them or something like that, no bacteria contamination, you can put that in the trash can. If it does have contamination, meaning bacteria, it should go in the biohazard container if it is plastic. And that's all we use for pipettes in the lab. You have an assigned drawer if you were actually going to a lab. All right, so where you put your books and your code, and if you need to change your shoes, you put your shoes in there, the little cubby hole or cupboard, and the door is considered outside the lab. You have to walk a few feet to enter the lab proper. Remember that. Any questions? You must follow all of these rules. Failure to do so would have a penalty, point penalty in the lab. And since we're an online class, I won't be checking that, but you should just know how to do the safety procedure, and you will be quizzed on it. All right, so today we're going to talk about Lab 1, lab safety training. Remember, you must turn in this lab as well as Lab 01, the lab we do on Thursday, by Friday at 8 a.m. Otherwise, you'll be dropped from the course. Any questions about any of that? Remember, you have the online plagiarism quiz, which is due at 11:59 p.m. this Saturday. To take the quiz, you must finish the plagiarism tutorial. And you must get 100% on the quiz in order to get potentially 100% on the infectious disease project. If you do not get 100% on the plagiarism quiz, you get 0 on the 100-point infectious disease project. Any questions about any of that? All right, let's go to the lab. I think I can close that down. I think that's the one I want, but let's see. Don't see it. All right, I don't have either highlighted, so I have to go through it. Ah, so lab module 0, lab safety for microbiology. Upon successful completion of this lab, students should be able to distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab, and understand how to safely dispose of contaminated materials. You know, that might be for the in-class lab. So let me go take a look at the other one. Yeah. Microbiologists are expected to observe a number of safety rules and procedures when they're working in the lab, especially when they're working with a pathogenic microorganism. While you're not going to be actually performing any activities in the lab, we do expect you to understand these activities and procedures. So you should wear proper attire when you come into the lab. You must have a clean lab coat that covers your lap when sitting. It must be buttoned up. You must have gloves on. You must have closed-toed shoes. And if you're working with a splash hazard, you must wear goggles. Note, most of the time in the microbiology lab, we will not be working with a splash hazard. So most of the time, you will not need to wear goggles. Long hair should be tied back. No food or drink in the lab. No electronics in the lab. No personal items in the lab unless you're wearing it. And that includes your cell phone. All personal items must be stored in the cupboard or cabinet or cubbyhole next to the door. And the door and the cubbyhole are considered outside the lab. Personal items that are brought into the main part of the lab will be confiscated if they should not be in the lab, and they will not be returned to you until they've been sterilized, and that would destroy any electronics, except maybe a waterproof watch. There's some questions about what you should be wearing in the lab, and this lab module does not really tell you, but you can answer it from looking at this picture. Okay, and she's wearing personal eyeglasses. Those are not goggles. Oops, trying to blow that up and it didn't blow up. Let me see if I can blow it up here. So to answer some of the questions, you may need to study this picture. All right. And it's trying to show she has to wear the lab coat, gloves, and then her hair has to be tied back. She might have a hairnet on, which does the same thing. Oh, come on. Can't shrink it down that way. Let me go this way now. And that just blew it up. You must disinfect your work area before you begin any work and then after you're done. Here's an actual picture of our microbiology lab. And somebody would be working right here. Somebody else will be working right here. Discard waste in the appropriate receptacles, regular trash that is not contaminated, and no sharp objects, no broken glass, no glass of any kind goes inside the trash can. And that would be something like gloves that are not contaminated, paper towels that are not contaminated. Anything that is contaminated goes in the biohazard bag. And I sometimes call that the autoclave bag because that's how we sterilize it. This will be pulled out, put in the autoclave, which will kill any microbes, and then it's thrown away in the regular trash. Swabs and slides and cover slips each have their own container and they're labeled, and they have disinfectant, and you put the swab or the slide or the cover slip in the container. The swabs and the cover slips will be just discarded, but the glass slides will actually be make sure they're sterile, make sure that the disinfectant's enough to cover it. They'll just add more if there isn't and let it sit for 15 minutes. And then they will wash it and then reuse the slides. If you do not know where to dispose of something, please ask your instructor. Report all spills and accidents immediately, no matter how slight. I've already given you the procedure, so I'm not going to give it again. No mouth pipetting. No putting anything in your mouth. And actually, I'm somebody like Jack Benny. I like to touch my face. And so whenever I'm in the microbiology lab, I try not to do that. Although the face isn't a mouth, but it's not something I want bacteria on. I already mentioned when you're not using the Bunsen burner, the bacteria incinerator, they should be turned off. Realize that the bacteria incinerator, it does take about 5 minutes for it to heat up, so then you can use it. You should be familiar with the safety equipment. We have a fire alarm, so you can push that if ever there's a fire. A first aid kit, puts that white thing up there. This is a blanket in case somebody needs to be smothered with a blanket to put out a fire, or what else would you use a blanket for? I don't think you'd use that if you had a chemical spill. I'm not sure what you'd use though, but there's a blanket for you. A deluge shower, if you got chemicals all over yourself and you want to rinse it away, stand it in the deluge shower. Fortunately, I've never had a student have to do that because if you do that, the water just goes on the floor. And it's true we have a drain on the floor, but it makes a mess on the floor. An eye wash station, I've only had actually a visiting scientist need to wash that where they got something in their eye. And I forced them to use the eye wash station. And then a fire extinguisher, which I've never needed to use on the lab. And there's a button there, but I don't even know what that's for. You don't need to know what that button is. All right, so the lab procedures and lab safety rules, I've already given you that, so I'm not going to go in it again. But generally only permitted students and staff are allowed in the microbiology lab. And we do run the microbiology lab under biosafety level 2. That's why you need to wash your hands before you enter the lab, put on your lab coat, put on gloves, and if you have a splash hazard, you put on goggles. And then before you leave the lab, you do the reverse. That's a biosafety level 2, as well as you need to clean the work area before you begin and then before you leave. Never take cultures out of the lab. Assume each culture is a biohazard, and don't bring in any cultures unless you get it approved by me. Discard of old cultures in the following ways. Agar plates will be in a plastic container with agar, should be put in the, assuming it's had microbes in it, should be put in the biohazard bag. Glass slides, cotton swabs should be put in the appropriate glass container, and it'll have disinfectant in it. The cover slips will have a separate container. Put the cover slip in that container. Pipettes should be in the appropriate labeled waste bin. And because we use plastic pipettes, if you have microbes on it, that should be put in the biohazard bag. If no microbes touch the plastic pipette, you can put those in the regular trash can. Glass test tubes need to have the label removed and put in the discard test tube rack, which is in the back of the lab. You don't really need to know about supplies in the lab because we're not going to go into a real lab, but we do have supplies available for you if you were to come into a real microbiology lab. And remember that where you put your books and the door is considered outside the lab. You need to walk a few feet before you get into the lab proper. Any questions about the lab? If not, didn't know I changed that, but let's go ahead and save that. Let's go ahead and talk about the worksheet. If you see page 1, which I think I deleted, you can skip it and go on to the questions on page 2. This has the questions shown to you. The other questions aren't shown unless you print it out. So answer rule 1 in the lab. What do you always wear for safety before you enter the lab? This is before you open the door and enter the lab. This is not directly stated in the lab, but it is shown in the picture. And then rule 2, after you enter the lab, name 2 items that you must wear to keep yourself safe while you're inside the microbiology lab. And we're talking about in the lab proper. And then 3, you need to look up a safety data sheet online, and it discusses chemicals in the lab. And the safety data sheet describes what? So provide at least a phrase for each answer, meaning a one-word answer is not acceptable. And your answer should be general to any safety data sheet. It should not be specific, like giving me the information, the safety data sheet, for example, ethanol. Okay, so it should be general for any safety data sheet. Any questions about that? All right, rule 4, know the location of your safety equipment. You don't really need to know the location, but you must name 5 items of safety equipment found in the lab. And that was in the lab module, so you should be able to get that easily. And then rule 5, name 2 items that you can take into the lab. And then the next question is, can you eat and drink in the lab? So, by the way, these asterisks tell you how many answers you should be giving. Don't erase the asterisks because that'll only confuse you. It won't confuse me. And so if you only get like 4 safety equipment, you'll get a -1. One of them is not correct because you're supposed to mention 5. And so name 2 items, 1, 2, and then can you eat and drink in the lab? And that's just a yes or no question. And then 2, list 1 item of laboratory attire that you must wear in the microbiology lab. And 3, list 1 item belonging to you that is allowed to be brought into the microbiology lab. Okay, and then list 1 item belonging to you that is not allowed in the microbiology lab. And then 5, it just reminds you this must be submitted by 8 a.m. on Friday in this folder for worksheet lab 00, or you will be dropped from the class. I'm going to use this for measuring attendance for students attending the lab. Any questions about this worksheet? If not, go ahead and start answering it. I will be here until 9:20 or the last student logs off. And generally, that'll be the last student logging off. All right, no questions? I'm going to stop the share and stop recording, but I will be here. There it is. Boy, this is a little